Okay, so let me do a brief introduction of Amy Orsborn, who is a assistant professor, still, I guess, going up for tenure this year at University of Washington, as well as, I think, with an appointment at the Primate Center there as well. Amy is working in the domain of monkey neurophysiology with a specific interest in brain-machine or brain-computer interfaces, I think with a very interesting focus on the closed loop nature of such control. And so she's really, I think, one of the doing some of the most interesting work in this space about how individuals can learn to work with such interfaces to optimize their performance. And today she's going to tell us, I think, mostly about one of her lab's recent preprints on this issue. So thank you, Amy, for joining us, and we look forward to your presentation.
Great, thanks. Yeah, thanks so much for the invite. Really looking forward to talking with y'all about this work. And so, like Andrew said, I'm mostly going to be focusing today on this preprint that's led by my grad student, Pavi Radeswaran, in a really close collaboration with people at University of Montreal, specifically Alex Payer and Guillaume Lajoie's group. So just want to sort of acknowledge them right at the front because this is all their hard work. Okay, so one thing I think is really interesting about motor learning is that we learn in all sorts of different ways, and it leads to lots of different changes in our brains. So for instance, there have been some really interesting recent observations that potentially we can adapt really flexibly and quickly, not by rewiring synaptic connections in the brain, but by using the flexibility of having so many neurons connected in complex networks to actually have population level mechanisms of learning. And you can imagine that this is really powerful in the motor system because there are so many cortical and subcortical areas that actually contribute to shaping our movements. And we, in fact, see as people learn motor skills that there's sort of very distributed changes across large areas of the brain. But on the other end of the spectrum, there's also really clear work showing that, motor learning and refining a skill can actually lead to incredibly targeted changes in neural plasticity. For instance, with specific spines forming as mice learn to master a skill, and you can actually go in and lesion particular spines, and even with small perturbations to that synaptic connection, you can actually lead to behavioral deficits, for instance. And so this sort of broadly is fitting with evidence suggesting that sometimes there seem to be really targeted changes reflecting some kind of credit assignment where, errors in a particular motor skill are targeted to specific precise synapses. And so we're kind of very broadly interested in this large question of how in the world do these single neuron and population level mechanisms of learning kind of ultimately interact to shape our motor behaviors. And this is a really tricky question to get a handle on, in part just because the system is so complex. And so one of the approaches that we've taken to this is to actually use engineered systems like a brain-computer interface to simplify the problem, because it specifically lets us define a mapping of particular neural signals in the brain directly to behavior, for instance, having neurons in motor cortex control the movement of a cursor. And so now, instead of having kind of distributed brain areas contributing directly to movement, we can go in and specify that only neurons in the primary motor cortex are going to control movement. And then even within motor cortex, we can go in and more precisely say exactly which neurons, the ones I've colored in purple here, are going to ultimately have direct paths to influence the cursor. And so for this talk, we're going to call those neurons that directly connect to the move the cursor readout neurons. And those are in contrast to other neurons nearby that might be right next door in motor cortex, but don't have kind of a direct connection to the cursor. And we can define that via our decoder algorithm. So the readout neurons, their firing rates are directly put into an algorithm that then does sort of a dimensionality reduction to generate a two-dimensional velocity to control the movement of the cursor. And so now, for instance, with this setup, you can start asking questions about, are there changes that are targeted specifically to the readout neurons versus the non-readout neurons, or are there changes throughout the whole population? And so we're not the first people to be interested in using BCI to study learning. There's a lot of really wonderful work in this space, but I want to kind of highlight that I think there's roughly two categories of how people have used this. So one is using BCI to really precisely apply perturbations to study things like adaptation. So Aaron Batista and Byron Yu have done really wonderful work that's an example of this kind of approach, where you train monkeys to learn some kind of initial BCI mapping, and then you apply perturbations relative to that. Most of this work focuses on kind of really short timescales of learning, and importantly, it's always relative to some initial starting point. In contrast, the other kind of line of work that's more relevant to what we're going to talk about today, you can think of as kind of analogous to studying kind of de novo learning of a new sensory motor transformation. And so in this approach, the goal is to record sort of roughly the same neurons as best we can with electrophysiology over days, and then use the decoder to sort of precisely keep that mapping of neural activity to behavior the same, and then understand how the animal learns that transformation. And so here, there's not really kind of a relative baseline necessarily in quite the same way. And we're also focusing on kind of slightly longer timescales. And so one really interesting observation that came out of Karnesh Ganguly's work with this approach is that you see sort of the formation of something that looks like a motor skill. So as animals practice with this sort of even arbitrary transformation of a particular decoder, if you look at the percent correct over time, you see something that looks like kind of a classic motor learning curve. And animals kind of consolidate this skill where they pick up where they left off on the previous day and kind of can immediately have high performance in these late days. And if you look at what's happening in the brain, this corresponds with sort of the formation of a relatively stable neural representation. Here, this is quantified just by looking at sort of how similar are the tuning curves, for instance, in motor cortex over time. And so as people were doing all of this work with fixed decoders to study questions of learning in parallel, engineers are actually doing a lot of work where they're doing something quite different and actually purposefully adapting the decoder to address kind of important goals around getting these devices to work for kind of real therapies. And so, for instance, the goal of these sort of adaptive decoders is to actually assist people in kind of that learning process rather than just giving them something that they have to try to learn. For instance, you can have someone using a BCI in closed loop, and then you look at mistakes that they make and you use that to change the algorithm. So that, for instance, if I generate a pattern of neural activity and my cursor goes up, but I meant to go to the right, you can then change the decoder so that the next time they generate that same pattern of neural activity, now the cursor will go in the correct intended direction. And there's lots of different approaches that have been developed for this kind of thing, including a lot of my own work. But I want to kind of highlight sort of some of the advantages of this. So this can potentially sort of help with that initial calibration phase. So rather than giving someone a decoder that takes them, many weeks to master, you could potentially sort of give them something that they can learn somewhat more quickly with this initial calibration step. But it also deals with other practical challenges where, Karnesh was doing these really heroic experiments where you try to maintain exactly the same neurons over time. That's not actually going to be practical in a real world BCI. And so you're going to have to try to adapt your decoding algorithm as your measurements change over time. And so because of some of these advantages, this has actually become a really common practice in most clinical BCI applications. And I would, really argue that in reality, most likely a lot of the ultimate technologies in this space will likely have some kind of adaptive decoding element. And so this is useful for an engineering perspective, but it actually potentially makes this really quite complicated in terms of the system, because you can think about, we now have a decoder that's trying to assist the user, but the user is potentially also learning at the same time. And so I'm very interested in this question of how do these two things interact? And so we did some studies to try to see, does the brain actually learn alongside an adaptive decoder at all? Because you could imagine that maybe the brain just kind of lets the decoder do all of the work, and then doesn't actually adapt very much. So we did experiments with a monkey where we had them control a computer cursor. So the velocity of the computer cursor was controlled with activity of motor cortex. And then we adapted the decoder for two specific reasons. The first was that initial calibration phase. So we would try to sort of assist them and give them a decoder that worked somewhat well at the beginning. And then we would also use this to deal with measurement drift over time. So anytime we lost a neuron, for instance, in our measurements, we would swap a new one in and then adapt the decoder. And what we found, interestingly, is that you see learning curves in this approach, suggesting that the user actually learns kind of alongside the algorithm. So the algorithm, for instance, here sort of boosted up performance to about 50%, and then the animal kind of gradually adapted and learned how to control this decoder. And then these sort of intervening times where we're occasionally changing the decoder to deal with measurement instability doesn't disrupt that overall learning trajectory. And so we see evidence of neural activity changes kind of reflecting this learning. And interestingly, sort of similar to what Karnesh saw with his fixed decoder, it seems like the neural activity kind of becomes more stable over time. And so, so there's one other kind of key aspect of this paper that I want to quickly point out because I think it's very relevant to the paper that I'm going to tell you about today. And that is highlighting the fact that learning actually still occurs even when you have pretty high task performance. So one interesting thing that we did in this study was actually vary the amount of decoder assistance at the beginning. So these are three different learning curves. So just the percent correct on the task, one where we didn't adapt much at all at the beginning, and then one, for instance, up here where we adapted so that basically performance is at 100% correct at the beginning. And so then you can look at how much does neural activity change as a function of kind of how much the animal had to learn, right, within that series. And what you can see is what you might expect, that if the decoder has kind of already given you pretty good performance and there's not much room to improve beyond that, there's much less neural activity change compared to if the animal has to learn quite a bit. But the thing I want to point out is that this intercept is not zero. And in fact, neural activity does still change in these cases where performance is really high. And just to kind of belabor that point a little bit, I'll show you an example of what is actually happening and kind of give you some intuition. And it's because even if task performance is high, the animals actually continue to really refine their ability to make these movements. So for instance, percent correct here has saturated at 100%. And in this example, we're actually adapting the decoder every day to make sure that it's the best it could possibly be. And even in this example, what you see is that the animals are continuing to get better at the task, generating more trials per minute and more accurate, sort of straighter reaches over time. And this isn't just because we're kind of adapting as much as possible and maybe we've trained the algorithm on more data, as you might expect. Actually, it's because there's changes in the neural activity as well. So this is examples of tuning curves of different neurons. These are just two different examples. Picking days over the session. And here's sort of a really striking example of a neuron that actually wasn't very tuned for this task, but gradually becomes tuned over time. And this is just kind of quantifying all that data of sort of how much the neurons are tuned over days. And so even when you have really high performance, the animals are still actually continuing to master this task over time with practice. Okay, and so given this phenomenon that we have algorithms that are adapting and changing, but the brain is also clearly learning alongside this, we were very curious to understand how do these two things interact? Because you could imagine that potentially the presence of this adaptive algorithm is going to change sort of what the brain learns or how the brain learns. And so in particular, this is interesting, again, going back to the point that the algorithm is often reducing errors, and we use errors to learn. So that may potentially be changing kind of the signals that the brain has available to learn. But also because, again, there's potentially trivial explanations for this, where if performance is really high, you don't have to change your neural activity that much. But the other sort of interesting question embedded in this is that the adaptive algorithms are also often just operating to maintain performance, right? So there's no change in the overall task performance from the kind of outside perspective, but the underlying actual sensory motor map has changed. And so does the brain actually care about this? And does that in some way trigger learning changes? And so what we did was revisit that data from the 2014 paper that I was just summarizing before to try to tackle this question. And so one of the places that we started was actually with sort of looking at population level structure in the motor cortex activity. And this is motivated in large part by kind of really nice work. Much of the recent work in motor cortex kind of really focuses on this observation that neurons don't work in isolation. They actually have really interesting coordinated patterns of neural activity. And often that once animals are doing well-learned behaviors, you see this really clear low-dimensional structure that emerges over time in motor cortex. And so does that same thing happen when we have our adaptive decoders was kind of a simple starting point. And so we started looking at this with a approach to measure sort of the dimensionality of the neural population representations. And we did that with a metric called the participation ratio. And basically what you can think of this metric as doing is kind of describing the degrees of freedom of a population. So if this number is 0, the neurons are fully correlated. And if it's 1, they're fully independent. So we can plot this sort of participation ratio metric for, this is just an example learning series. And what you can see is that in this case, we actually, if anything, have kind of this increase in dimensionality that's happening over time. And this, again, is sort of in contrast to what you might expect, because most previous evidence in motor cortex literature suggests that as you learn a motor skill, you tend to sort of form more stable representations that also become kind of more low dimensional. And in fact, we went back to double check that this was true in BCI. We went back to Karnesh's data with fixed decoders and analyzed their data with our same approach, and in fact saw that in that case with a fixed decoder, you do actually see sort of this gradual reduction in the dimensionality of the neural population for their data. And so these are just example series, but just to summarize, if we have many different series where the animals are learning these adaptive decoders, and what we saw is across 2 monkeys and across all these different series, either the dimensionality was not changing, so it wasn't getting smaller, or if anything, it was increasing. And this was in stark contrast to the data that we had from previous studies with fixed decoders. So something is going on that is sort of somewhat surprising in this, suggesting that the adaptive decoder is sort of manipulating things in some way. And we got further evidence suggesting that the adaptive decoder is changing something in the neural representations by noticing that if we look at sort of how much this dimensionality changed, so just taking this metric, but now plotting it as a function of how long the series was, we saw kind of this weak but clear correlation. So one thing that happens when a series runs longer is that there's more likely to have been more interventions where we adapted the decoder. So we wanted to try to see, is there something where when we go in and manipulate the decoder, does that seem to drive the changes in dimensionality? And so here's where I kind of have to unpack a technical detail of this experiment, where there actually were 2 kinds of adaptive decoder changes that we were making. So if we go back to sort of that transformation of neural activity to the cursor, in some cases, all we were doing was changing the weights of the decoder. So the readout neurons would stay the same, but we would simply change the relative weighting of how they contributed to the velocity of the cursor. And in other cases, we were dealing with this measurement instability problem. And so we were actually changing the identity of the readouts and then correspondingly changing the weights of the decoder as well. And so we split up our decoder perturbations into the ones where we only changed the weights and looked at the participation ratio sort of right before the change or after the change. And here we didn't see kind of any statistical difference. But if we looked at the decoder changes where we also changed which units were controlling the cursor, that's where we saw sort of an evidence that that seems to be what's driving these kind of increases in dimensionality. And so it seems like something is going on where once you introduce an adaptive decoder, you're kind of changing the neural dynamics of learning that we're seeing, definitely from different than what we might have expected on initial hypothesis. But also this highlights that we kind of don't have a clearly sort of static low dimensional manifold structure to work with in this data set. And so we decided to sort of, and then the last thing is that it also points out that sort of the identity of the units may really matter because, for instance, these changes in the neural population dimensionality seem to be linked to when we change which neuron is controlling the cursor. And so that led us to kind of shift gears a little bit to actually take a different approach that in many ways is actually slightly more old school, where we actually just look at kind of the individual neurons and how they relate to movement. And so the approach that we took to do this was using an offline classifier. So just to be completely clear, this is another decoder, but it's a decoder that's not what we were using to predict the actual cursor movements. And so we take the activity of neurons, we put it into a classifier and say, can we predict which target the animal was trying to reach to? And we can do that for the different neural populations, for instance, readouts versus non-readouts. And so what's really useful about this approach is that it helps us get a sense of which neurons have information about the task and how that might be changing over learning. But in particular, one of the reasons that we ultimately settled on this approach is because when the animals are learning, things like the kinematics and the details of their reaches are changing. And so it's often easy to see changes in neural activity that actually just reflect the fact that the behavior changed. And it's hard to disentangle those two. What's really nice about this approach is that the targets are things that are fixed, and those are always the same. And so it kind of controls for this. And I'll kind of touch back to some other, technical details about why we think this approach is particularly powerful towards the end as well. Okay, and so with this approach, one of the things we can do is just double check that this is giving us reasonable capturing sort of reasonable effects in the data. And so, for instance, we can look at the coefficients of our classifier. So this heat map representation, basically neurons are on the y-axis here, time bins are on the x-axis, and then the color tells you basically if that neuron at that particular time has information about the cursor direction. And so this is just an example map early on. What we would expect, given that we know that neural activity seems to become more stable, is that this might go from sort of a weird salt and pepper map that's changing a lot to something that becomes more stationary. And that is in fact what we see. So if we look at sort of these different early maps, they're all over the place. But once you look late in learning, there's sort of this pretty stationary map of classifier coefficients that are similar over time. And we can quantify that, for instance, by just looking at how correlated are these two maps and how does that relate to, for instance, the performance improvements. And here's just one example series showing that kind of the maps are stabilizing as the performance in the task improves. So this is in some ways just replicating things we had already known from the 2014 paper, but just with some different methods for kind of consistency checks. Okay, and so now we can look at questions of how is this information about the task kind of distributed across the neural populations. And here we can really take advantage of this BCI paradigm to look at, is it specific to, for instance, the readout populations or the non-readout populations? And so we can use our classifier to look at how well can we decode the movement of the cursor over days. And so if we use all the neurons, what you can see, and as you might expect, is that we kind of gradually improve our ability to predict the target location as the animal gets better at the task. But if you break this out into which different neural populations basically this increase in decoding performance is entirely coming from representations within the readout neurons. So the non-readouts have information about the task, but they don't have nearly as much as the readouts. That's somewhat expected because they're not directly controlling the cursor, but also the non-readouts basically don't change at all. So the learning is always targeted to the readout neurons, and we can quantify that kind of across all of our different series and our two animals, where we kind of clearly see that there's the learning increases kind of only happen in the readouts as compared to the non-readouts. And so, so this is consistent with sort of some kind of targeted changes specific to the readout neurons in our data. But as we looked kind of at these example coefficient maps, we noticed something really striking, that it's not just that all the readout neurons, for instance, become very informative. It actually looks like there's really just a small subset of readout neurons that seem to have much information about the task. And so we wanted to kind of drill into that a little bit more. And so we did that by a variation of our classifier analysis, where we add neurons basically over time. So what you can do is take each individual neuron as inputs to your classifier and say, how much can I predict about the task from that neuron? And you can get kind of a ranked list of most informative to least informative. And then you can gradually add those to a neural population over time to build this sort of curve of how well can I classify as I add neurons to my population. And so when we do this, what we find is that this curve interestingly kind of shifts over time. So early on, we have to add quite a few neurons before we get to kind of the saturation of how the maximum decoding performance. But late in learning, after we add just a small number of neurons, in this example series, about 5, we've kind of hit our saturation point. And this phenomenon was really robust, where if you look at kind of how many neurons do you need to predict the cursor position early versus late, that number kind of always was going down. And so we call this kind of a compact representation, because it seems like the information is kind of only represented in just a small number of neurons. And so, but to really kind of make this point, to try to fully understand this, you can imagine that maybe actually this is just a really efficient representation and I could grab any 5 neurons, right? And that would work. So we wanted to double check, is it actually a specific 5 neurons? And we did a variation of that neuron adding curve, but where now we actually use every possible combination of neural populations for a given size of neuron. So in this example of 2, we would take all possible pairs of neurons within our 16 readout neurons and test how well we can classify with all of those. And so that's kind of this big cloud of data points that you see. And then what we've done is, if you have those sort of top 5 neurons that we identified before with that previous analysis, then we colored things in those dots in purple. And if those are absent in the population, we've colored those dots in orange. And so this is what's happening early in learning. And you can see sort of that these two populations sort of overlap. So there might be some mixing of information between different neurons in the brain. But if we look at what happens late, these two populations separate. And so what this really means is that the improvements in performance are happening because of changes in those top neurons only, right? So that is what's kind of driving this shift in decoding performance. And so we can quantify that again across series just by looking at sort of the D prime, basically the separation between those two populations. And in all cases, that grows over time with learning. And so it does really seem to be that just a few neurons are what's driving this learning effect. It's very targeted. I also want to kind of quickly mention sort of address one possible concern. So, we are with this analysis kind of simply classifying target identities. And we did that for strategic reasons because those are consistent over time in the task. But as I pointed out to you earlier, some of the improvements and that animals are making in this task are actually about kind of the details of the kinematics. And so you might be concerned that maybe this actually misses sort of kinematic details that are relevant to skill. And so to try to address this, what we did was use our classifier to rank these neurons from sort of most predictive to least predictive. And then we did a version of the sort of neuron adding curve, but where we actually predicted or reconstructed the cursor trajectories with the decoder that we know, right, to try to see when can we accurately reconstruct the cursor. And so this is an example of that curve. There's very boring, uninteresting reasons as to why X and Y are different here. But the key thing here is that as we add neurons, once we hit that number of, again, for five for this example series, we basically don't need to add any more neurons to accurately reconstruct the cursor. And so this is sort of capturing something about the cursor trajectories, even though we're ultimately looking at just simply target identities. Okay, so to sort of summarize where we are so far, we see this interesting evidence that adaptive decoders alter kind of what we might have expected in neural populations, where we have this non-decreasing dimensionality. And this might be related to sort of this phenomenon of swapping the neurons. And when we turn to unit level analysis, we see this really kind of interesting thing where the representations become really compact. And this is not something that had been kind of previously observed. And this left us scratching our head because it actually seems slightly inconsistent with the idea that the population is not becoming lower dimensional. And so this sort of raises two kind of questions that I'll want to address in this sort of back piece of the talk. So the first is all of these evidence sort of show interesting phenomena that we're seeing in our data, but we don't actually yet have direct causal evidence to show that this is because of the adaptive decoder, right? So we want to try to get at that question. And then the other is, how do we actually ultimately reconcile these two phenomena that we've seen described at different levels of neural activity? Okay, so first, let's get at the question of is the adaptive decoder kind of causally contributing to things? And so this, unfortunately, is really difficult to do experimentally. If I could take the exact same monkey and do two different experiments with it, that would be wonderful, but there's lots of training history, dependent effects, and other phenomena that make this very difficult. And what you would actually need to do is use tons and tons of animals to kind of rigorously get at this question. And so instead, what we turned to was simulations. And so Guillaume's group has done some really wonderful work developing artificial neural network models of BCI learning. So this is Alex's work. He's built a really great model where you have an RNN. You can train it to actually initially control an arm. And then you can, once the network is trained to control that arm, you can then do a BCI experiment where you say, here's just a few neurons within this RNN that are now going to control this decoder, and ask the model to also learn how to control this BCI cursor. And so the model, I won't go into a lot of the technical details, but it's trained with a biologically plausible version of gradient descent called reinforce. It learns continually, which is actually really important and kind of a key technical innovation when you have an adaptive decoder, because you don't necessarily want to sort of train the model at certain times and then fix it. So this better mimics kind of what might be happening in our experiments. And then Alex has also done a lot of really great work kind of using these models to make sure that it really captures a lot of previous experimental observations of BCI learning, giving us sort of some confidence that this might be capturing interesting phenomena, new phenomena that we see in our data. Okay, and so Alex can train the model to learn how to control arm movements, and then ask the model to learn the BCI. And at the beginning, for instance, it's not very good. It generates these kind of wonky trajectories, but the model can eventually learn how to control the BCI. And then Alex can now do the experiments that I can't, where he can take literally the exact same network and run the experiments with different levels of adaptive decoding to then try to really understand how the model interacts with the adaptive decoder. And so the first thing is just to double check that adaptive decoding works well and you don't disrupt task performance. So this is task performance over time, sort of days, quote unquote, in simulation. For different levels of adaptive decoding, there are some really interesting phenomena where if you actually start cranking up the adaptation too much, you actually start to hinder performance. This is consistent with experimental data as well. Yeah, there's some interesting nuances here in the model that the model kind of nicely captures. And so now we can look at what is happening in the neural representations that are learned by the networks with these different levels of adaptive decoding. And so we basically did a version of the analysis that we did with the experimental data, where we look at sort of the performance as we add neurons into the simulation. And so these two curves, the dashed is early, solid is late, for a fixed decoder. Basically, these two curves, there's a lot of variation in here, but what you can see is that the main takeaway here is that these two curves kind of roughly overlap. But if you start adding in even any small amount of adaptive decoding, you see that these early and late curves start to diverge. And so what this is sort of equivalent to that shift that I was showing you before in the neuron adding curves in the experimental data, where basically the model is able to perform the task well with a smaller number of neurons. And so we're getting this compact representation in the network as we introduce the adaptive decoding. There's also some really wonderful analyses that Alex has done that I won't touch on for the sake of time, but where you can see that the network weights, for instance, across the units are clearly influenced by the adaptive decoder. So it's not just the readout neurons, but all of the neurons seem to be influenced by the adaptive decoder. Okay, and so in the last few minutes here, I want to kind of move from this question of sort of, is the algorithm causally impacting this, which our answer seems to be yes, based on simulations, to now kind of try to reconcile these seemingly disparate observations about unit level representations versus population level perspectives. And so what we did was first just ask a simple question of, if we, instead of looking at single neurons, look at this neural activity from sort of a population mode perspective, right? Is that also compact or not? So now we're going to do our same classifier analysis, but instead of putting in individual neurons, we're going to first put the neural activity through a dimensionality reduction like PCA to get different modes, and then put those into the classifier. And so interestingly, what we found is that if we now build that sort of mode adding curve, right, where we're adding principal components and looking at how well we can classify, what you see is that early, we have to add quite a few principal components, but late in learning, we actually just need to add two. And then there's sort of relatively little information about the task after that. And this was true, again, across all series, that sort of the number of principal components that you need decreases over time. And so it's not just that information is becoming compact when you look at it at the level of single neurons, it's actually also becoming compact at the level of kind of population modes with learning. So now the question is, why is that not decreasing the dimensionality of the neural activity? And so to get at this, we looked at the variance explained by the population, by the different modes of neural activity. And so this is just one example series, and you can see sort of this change, early versus late, that is consistent with the fact that the neural activity is getting higher dimensional, right? So early on, actually, most of the variance is in the first couple of PCs. Late in learning, the variance is sort of spread out a little bit more across different PCs. But now what we can do is look at how, rather than just looking at variance explained, we can look at the prediction accuracy. And so if we look at decoding power for each of these different PCs, what stands out is that where kind of the learning seems to happen is actually out here in the fringes in these modes that have relatively low variance. And so we quantified that just by kind of breaking up our PCs into the ones that explain the top half of the variance versus sort of the bottom 50% of our PCs that don't explain very much variance. And if you look at kind of the decoding power, the task information that's contained within those different sets of modes, you can see that it's actually kind of all in the bottom over here. So the low variance modes are actually what has task information about BCI. And so that kind of reconciles why we're seeing these two different phenomena. But the last thing we wanted to kind of get at is this because could it be that the computations are happening at the level of populations, or are they really happening at the level of single neurons? And this is a tricky question to ask, and we haven't kind of fully cracked it. But one thing that's really interesting is that we can look at, for those modes that are kind of very predictive of the task versus modes that have just a lot of variance in the neural activity, where are they getting their inputs from? Kind of what are the loadings, right, for different neurons? And so we ranked the neurons by kind of, were they very predictive about the task when we looked at that at the single neuron level? And then we looked at the loading of that neuron for different PCs. So for PCs that had a lot of decoding information, you get a curve that you would totally expect. Like if our analyses are working correctly, you would expect that neurons at the single unit level have a lot of task information, should also be projecting to these PCs that have task information. But what's really interesting is that if you look at the high variance PCs, they're kind of just getting information from some weird smattering of neurons that are both participating and have information about the task, and ones that don't. And so that's just one example series. We kind of quantified this across all series. And so what you can see is that clearly the top strongly decoding PCs are kind of preferentially getting inputs from these task predictive neurons, and the high variance PCs are getting some weird combination of inputs from a lot of different neurons. And so one way to potentially interpret this is that we have these compact representations at the level of individual neurons or at the level of modes. We can't really separate out where the learning is happening, but they occupy this really small amount of variance. And so they're kind of learning some representation that's like pushed out at the fringes of the neural representation in terms of variance. Okay. The last one other kind of quick thing I want to mention that I thought was really interesting is that all the things I've just shown you so far have quantified this just sort of across an individual series. So the animal would practice with a decoder for like 2 weeks, and we would look at changes early versus late. But we actually also ran these experiments over the span of, about 6 months, doing these different series with the same animal. And we see this trend that basically the number of neurons the animal seems to be using to do the task just keeps decreasing over series. And so that, I think, is quite interesting, and actually sort of potentially explains some of these data points where, like, for instance, basically there's 1 or 2 PCs that have information about the task, and they have like input 100% input from just a couple of neurons, which I find kind of super fascinating. But we can't make strong claims about it because that's a difficult experiment to do. Okay, so to try to sort of summarize here, we see that the brain learns alongside an adaptive decoder, and it sort of forms these stable neural encodings, consolidates some sort of motor skill. And in particular, there's sort of this really clear credit assignment where it seems like some of these changes that are happening are really targeted. And kind of our interpretation of this data is that, in particular, the adaptive decoding seems to kind of alter some of that process, and especially maybe altering that credit assignment, because we're getting kind of this hyper credit assignment where you end up with very compact representations, both at the level of single neurons or at the level of populations. And kind of you learn these really low variance representations. And so, this raises all sorts of questions that we talk a little bit about in the discussion of the paper of like, why is the brain doing this? And to be honest, we have some speculations, but I think this is kind of a big open question. You could imagine that maybe these are really efficient if you want to kind of store a very specific memory. And then, but the other thing kind of that I think this result highlights is that neural representations can change, right? Even when performance is high or doesn't change, and all you're doing is changing the sensory motor transformation. And what this suggests is actually that the brain is not just using task errors, right? It's likely also using some sort of internal signal to guide some of these changes, which I think is really interesting and we need to dig into more. Okay, and so I will sort of wrap up here because I think I'm about out of time, but I also want to just mention that I think this has really important implications for thinking about how we design these BCIs, because what it really means is that algorithms that kind of ignore plasticity could potentially be counterproductive, right? If these compact representations are not actually very beneficial, do we want the brain to be forming them? They're certainly potentially problematic from the perspective of kind of robustness of these devices over time, because if an animal learns a representation that uses 2 neurons and I lose one of those neurons, I'm really kind of out of luck. And so we've been working a lot on different kinds of frameworks for thinking about co-adaptive decoders. We have a recent new preprint that I'm pretty excited about that kind of is getting at some of that direction. Okay, and so with that, thanks so much to all the folks that actually did the work and all of you for your attention.
Awesome. Thank you, Amy. Um, is your audio working well there, Murdad? We're gonna open up to questions if you have a few minutes, Amy. Um,
Yes.
Yeah, can you hear us? Is it clear?
yeah, yeah.
Perfect.
We can't usually see you as normal, so if you have questions, go ahead.
Okay, I can start. So I had one clarification question for the first of all, thanks for the talk. And my question was for the adaptive BCI with the one that you changed the readout. What is the nature of that changing? Is it like completely taking out randomly a bunch of units at some point and putting them back maybe later on?
Yeah, great question. So no, it's not. These experiments were basically done very, not in a systematic designed way, but frankly, sort of more opportunistic. So it was just based on reality, dealing with realities of what was happening in the experiment. So if I was using 15 neurons, for example, as my readouts of my BCI, but I lost one of them, in the next day I come in and that unit is sort of not well isolated anymore, or I can't find a neuron on that electrode, then I would just take a different neuron and put it in. And so there's not kind of a controlled analysis for when those were done or how many neurons were swapped and so on, because it was all just based on what was happening and dealing with kind of practical realities. I do think there's some really interesting opportunities to sort of try to design more systematic versions of these experiments. We just can't get to them yet, right, with this current data set.
Thank you. So in that case, I was wondering if the results can be explained by a simple explanation like that you have like 2 or 3 really high quality units that they're just always there, but the other ones kind of drop and you replace them with the new ones. But the BCI over time just learns to rely on those like 2 very high quality units. And that's why we overall see that the number of effective neurons is going lower and lower.
Yeah, that's a great question. I do think there's some potential possibility of this. I don't think that's all of it though. So there's a few things that we have done to try to make sure that these aren't, for instance, just like random consequences of this like measurement drift that we're not able to control. So we have, in addition to this data from the animal doing arm movement tasks every day. And we can look at kind of some of the properties of the neurons in those cases. And we don't see a clear evidence, that sort of, oh, it's just the neurons that are like very informative about arm movement, for instance, that also then are useful for BCI, and unit quality kind of doesn't fully explain some of the phenomena that we're seeing. But I think you're definitely onto something in the context of this sort of reliability thing. So one of my interpretations of what's happening is basically a positive feedback loop where the animal kind of, for whatever reason, there are certain neurons that seem to be useful, right, for doing the task. The decoder gives those neurons high weights, and the animal kind of starts, increasing the contributions of those neurons. Then when you adapt the decoder is going to pick up on the fact that those neurons are useful and crank up their weights, and then it just like iterates from there. So it's almost like a version of overfitting, right? But it's overfitting via this interaction, right, between the brain and the algorithm. Yeah.
Thank you very much. That was a nice intuition.
Yeah.
I can jump on that question because I was interested in kind of the trade-off here in terms of generalization. It seems related to that last point, right? So, I mean, have you checked in this adaptive scheme how whether or not things just get much better maybe, but much more brittle to the experimental conditions at play?
Yeah, that's a really great question. So there's a few things we've done related to this. One, early on when we were doing these studies, we were really worried that we were just going to get them to be really great at the center out task and nothing else. And so we did double check that they can do like random target pursuit, for instance, and then also things like obstacle avoidance, where you give them a center out movement, but then you put something in the middle that they have to go around. And they can do all of those things just fine. So it does seem like they're learning some slightly more general mapping from neural activity to velocity as opposed to kind of discrete point-to-point movements. So that's great. But I agree with this is a very specific task of just controlling the velocity of a cursor. And so I think there's really interesting questions kind of getting at this point of what is the sort of reason for these compact representations around, is it some way for this to be really robust, to other different factors? And so some of the new projects that we are thinking about that we have new grants on is actually trying to do things where for instance, some of the early studies, we'd make monkeys learn to control this BCI cursor, and then we'd make them learn to do like arm movements plus the BCI cursor as like an extreme perturbation of that to get at kind of this question of generalization and robustness. And there's some preliminary evidence that kind of as they learn to separate those two tasks and the representations become kind of separate. And so, but that's all kind of, it's hard to make those claims rigorously with the data we have. And so we're kind of really interested in doing those kinds of assays with this and trying to understand, if you get them to learn a certain kind of representation, does that actually make it more robust to like combining with other behaviors or other kinds of perturbations and so on. Yeah.
Cool, thank you.
I have one question. Can you hear me?
Yeah.
So it's regarding the number of neurons involved in the task early in the learning and later in the learning. So I have one clarification question that like early in the learning, so the behavior could be slightly noisier and thus the neurons are noisier. So within each single neuron, you have less predictability power to like to see how much the task is encoded within a single neuron. So have you done some sort of like noise ceiling analysis to see if the noise ceiling, like the predictability power of the single neurons, how they differ, how the noise level is different across early learning and the later learning, and how this can contribute to the fact that the lower number of neurons are involved later in the learning?
Yeah, good question. So so you're absolutely right that kind of our ability to predict overall increases with learning, related to sort of the phenomena you were describing. And so, for instance, a lot of the curves that are actually presented in the paper, and you have to look at the supplements to see kind of the more raw data, are often normalized, right? So that you can kind of correct for some of these just general drifts in decoding performance. I think the things that make me somewhat more convinced that we're not just capturing kind of that property are the analyses that I was showing, the combinatorial things, where kind of you look at all possible combinations of neurons, kind of convinces me that there is something kind of very specific to those particular subsets of neurons, because in principle, if the brain is learning kind of some stable representation and all the neurons are getting kind of more consistent over time, then the decoding performance would improve for all of the neurons, but we're not seeing that, right? We only see it improve for kind of just a subset.
And I have a follow-up question. I mean, not follow-up, but like related. Is that the fact that so in machine learning community, there is this concept of like you drop out some sort of like weight during the training, and that result in like more distributed representation for the test, so that like more neurons are involved. And seems to be in line with the result that you said, that like the cross correlation of the neurons would increase during the learning with the adaptive algorithm, not with the fixed decoder. I was wondering about your thoughts on that.
Yeah, so you've actually sort of nicely described one of the aims of a grant we have with Guillaume, where we're really interested in trying to understand, can you do simple manipulations like dropout during training to actually change the representations that the brain learns to, for instance, make them more distributed towards a goal that may be more robust in some ways, right, than others. And so I think those are potentially really interesting directions to think about. And that's kind of, to me, one of the exciting kind of aspects of this finding to me is that, you know, on one hand, it makes it really complicated because we now have decoders that really kind of need to pay attention to what's happening in the brain. But the flip side is that it potentially opens up the possibility that really simple interventions, right, like software manipulations, can actually sort of shape plasticity, as opposed to having to like do electrical stimulation or some kind of other pharmacological manipulation or something like that. Yeah.
Thank you so much. Yeah.
I have one question. Can you hear me well?
Yeah.
Okay, so I was wondering what will happen if you change the number of free neurons. So, for example, making them so much lower or so much higher. Will the patterns of your result stay the same?
Yeah, I think that's a great question. I think there's two aspects to that I'll just quickly touch on. One is actually kind of newer experiments that we're planning on doing. We'll get at this a little bit, that all of our metrics in this paper are effectively inferences after the fact, and we don't know definitively that, yes, those neurons are the only ones kind of really contributing to behavior. And so what you would really like to do is actually online experiments where you like take out the neurons that you think aren't important and show that it doesn't impact performance and things like that. And so we are planning on doing those some of those kinds of experiments to follow up on this. But the other part, I think you raise a really interesting question about how much this is a function just of the overall population size. And I think I agree with you that it likely is. I don't have a good answer or like clear intuitions for this. One thing that's super interesting in the BCI literature is that it's clear that the number of neurons likely matters in terms of how animals learn these things. So like Mark Schieber's group has done some really cool experiments with like very small number of neurons, so controlling cursor with like 1, 2, 3, 4, 5 neurons, and they see that, for instance, it's like easier for the animals to learn transformations as you add more neurons, because you kind of increase the redundancy of the possible control space and things like that. And then we've done these experiments that are in kind of like the 20 to 30 neuron size range. And we see these like really interesting credit assignment phenomena and so on. And then there's people that have done experiments where they're like, we're just going to put in like all the Utah arrays we have and like, hundreds of neurons. And they often are not really clearly seeing the same evidence of credit assignment and so on. So there's some really complicated curve that like is hidden in the literature that I don't think anyone really fully understands yet. But I think it's a great question and something that I'm really interested in trying to understand a little bit better, because I think there's interesting ways in which it influences potentially the computations, right, that the brain has to perform.
Thank you.
Is there any more questions? I think we have time for one more. We promised Amy an hour. I don't want to keep her too long. Maybe one more question.
I have one.
Okay, go ahead, Marco.
Yeah, very, very, very simple one. I was curious about how the readout movement look like in terms of kinematics. Do they resemble the kinematics of like biological, like normal movement? Are they initially bumpy and then they become more like bell-shaped velocity profile?
Yeah. Yeah, it's a great question. I'm trying to see if I had slides on it. I don't, unfortunately. The thing I can quickly go back to, so they're definitely not quite as good as arm movements, but they get pretty close. So if wow, where is this? Okay. Million zoom windows. Okay. So these orange lines, I didn't touch on it. This is like what the animal does with their arm. So in terms of kind of simple metrics of like success rate and so on, they kind of get close, but I think there's no orange line here because the movement error is much, much lower in arm movement, right?
Okay.
So they are not quite as straight. They also have some really interesting phenomena of that I think are related to the fact that this is visual feedback only, for instance, and you have no proprioceptive or tactile input. They look very feed forward, for lack of a better term. So like I sometimes describe it as the monkeys are like learning how to push a slow moving boat, right? They sort of like generate a ballistic movement, and then they also, because our task requires them to like stop on a dime and like hold at this particular target. So my intuition of what they're doing is learning how to generate like a perfect, bolus of velocity to shoot the cursor to the target, have it slow down in the target, and then they pull it back towards the center. So
Okay.
they're kind of doing this like push-pull thing. Yeah. And you see some interesting phenomena of that because if they know they're overshooting, they'll like wiggle before they get to the cursor because they're trying to slow it down, so that they
Okay.
stop on the target and things like that. Yeah.
Thank you. Thanks.
Yeah.
Amy, will you indulge us one more? I think there was one more in the
Yeah.
room there. Okay, one more.
Yeah, sure. Yep.
So just a quick question. Thank you for the talk. I have a question about the results you showed that you looked at the decreased variance across the days in the previous data and in your data with an adaptive decoder. So I mean, this is just a descriptive, more naive view of that result. Would be that given a fixed decoder, it takes, for example, 10 days for a neural system to go down in terms of variance. But then when you change the decoder in like day 7 and then day 9 and then like before that 10-day point, you won't let like that dynamical system to go down in terms of variance. So I wonder if that would explain the result here that the variance never goes down because the changes are shorter than that, like the dynamic, than that duration that it takes for the variance to go down.
Yeah, that's an interesting point. I certainly think there is something one kind of obvious observation in this is kind of exactly to your point that with the fixed decoder, there's kind of much more significant learning, right, that's happening kind of in that early phase. And if you look at this plot and piece together some of the other plots, I think you're onto something that like this phase where things are somewhat flat is kind of when performance is improving a lot. And then once they're kind of in that plateau is when they kind of consolidate to some more stable representation. Yeah, and I do think there's some aspect of this kind of within the data, because also if you look at, we can't study this because we don't have enough data from the fixed experiments, but if you look at absolute numbers, there's also some kind of like interesting phenomena going on here. But I think what my interpretation of the adaptive decoder is that there's potentially slightly less exploration, in this in the adaptive decoders because you kind of have something reasonable, right? And they're not having to improve performance quite as much. But once you're kind of out in these plateau phases where performance is definitely good, you, if anything, start to see these increases and drifts, and those seem to be very tied to these like perturbations that we're doing where we're just maintaining the performance over time. Yeah.
Thank you.
Awesome. Okay. Well, Amy, thank you very much for the presentation. It's really a fascinating field right now that's exploding in so many cool directions, and it was great to see kind of the latest thinking on how these things are going. So yeah, everybody to join me in thanking Amy virtually or in the real world. Yeah, thanks again.
Awesome. Thank you so much.